thank you for joining us. I'm your host, Dr. Jay Feldstein, and today we are joined by Dr. Brian Balin, Director of the Center for Chronic Disorders of Aging and Professor of Neuroscience and Neuropathology, along with Nikki Schultek, Founding Director and Research Assistant Professor of the Pathobiome Research Center at PCOM. Nikki founded the Pathobiome Research Center in 2025 as a result of a near decade of collaboration with Brian, exploring the connections between infectious processes and chronic disorders. Their work brings together scientists, infectious disease specialists, and healthcare providers to advance research and education in this emerging field, with the goal of improving how we diagnose, prevent, and treat conditions linked to infection. Today, we'll discuss their ongoing Alzheimer's disease research, what excites them most about recent scientific advancements, and their vision for the future of their research center. Welcome, Brian and Nikki.
Thank you, Dr.
Thank you.
Feldstein.
So, Brian and Nikki, I got a couple questions for you. What sparked your initial collaboration, and how did it evolve into Nikki establishing the Pathobiome Research Center?
So, Jay, I reached out to Brian during one of the most difficult periods of my life. Going on 11 years ago, I was really sick. And that was actually what prompted me to reach out to Brian. I had found his research on this stealthy bacteria, Chlamydia pneumoniae, in relation to Alzheimer's disease. And I had this incredibly difficult period where I was diagnosed with one autoimmune or inflammatory condition after the next. And one of my terrifying symptoms was issues cognitively, but also neuropsychiatric symptoms and neuroinflammatory symptoms. My doctors were working me up and ruling out multiple sclerosis, but I had also developed a bladder pain disorder, joint pain, cardiac arrhythmia. I mean, literally, if you asked about a part of my body, it was inflamed. And so I started thinking about inflammation and these different systems of my body. And I noticed that the specialists I was seeing, and I had about a dozen, were incredibly siloed. And they would think about the one body part, prescribe medications to address the symptoms. But I was curious what was driving the whole thing. And I had found research by another researcher that was really passionate about studying Chlamydia pneumoniae bacteria. And that led me down the path of understanding that this very stealthy, common pathogen that nearly everyone gets exposed to, and it could become chronic, is linked with multiple idiopathic and very common and prevalent disorders, some of which I was diagnosed with, including asthma. And so this led me to begin in a very grassroots way, connecting. And I found a physician, Dr. Charles W. Stratton, who treated me for this infection, who had already known Brian. Right at that point, Brian, you had already known Chuck Stratton?
Yeah, yes, for 15 years.
So, Brian, I emailed you out of the blue one day, right?
You did, and then you called me as well, a cold call, and we started talking about this issue, and we realized that there's a commonality in what our approaches would be, and you from the patient perspective, and me from actually, coming into this research in a stealthy way of just basically asking the question of how could a microbe like this be involved with Alzheimer's disease?
Yeah, and Brian told me this really neat story of how he got inspired to look at the brain, right, Brian? You were working, I'll let you, you'll tell it better than me, but how did, I think it's cool for you to share how you ended up on the brain because you very much bridged silos, which is something we talk about a lot.
It's very true. So, I have a background, a bachelor's of science in microbiology. And then in my graduate work, I was looking at the blood-brain barrier and other barriers into the brain and realizing at that point in time that the work being done in the Alzheimer field, and this is in the 1980s, looking at atherosclerosis was now being linked to Alzheimer's disease. In the mid 1990s, after I had my own lab actually at a different institution, I was talking with a colleague of mine, Alan Hudson, and he's a molecular geneticist looking at chlamydial infections. And he mentioned to me that this one organism, Chlamydia pneumoniae, was being associated with atherosclerosis. So I just asked the question, could this have been an organism, or could this be an organism that's associated with brain disease because of migrating into the brain through the vasculature? So we decided to look in brain tissues that I had gathered over time because I was already looking at Alzheimer's disease. And we found evidence for that in these brain tissues of individuals that had died from Alzheimer's disease. Subsequently, we have been able to understand that actually our sense of smell seems to be our greatest risk because this is a organism that's a respiratory chlamydial organism, not the sexually transmitted form. And when we inhale this, it can then infect our olfactory neuroepithelial cells in our noses and then go directly into the brain. And I was studying that pathway in the 1980s, okay? And I have a paper in 1986 actually showing that a lot of molecules, a lot of pathways can be, or a lot of proteins, molecules of almost any sort that you inhale can actually get into the brain through this mechanism. Because Alzheimer's disease starts in the connections in the brain that are associated with our sense of smell. It's exactly where the disease starts. It became sort of like an opening of the window of understanding that infection going into our brains this way could actually lead to inflammation that could start this whole process. And to this day, we truly believe this is causative and can be causative for this problem in this manner.
And so, going off of what Brian said, at this point in time, I'm a sick person, right? So I end up getting treated with antimicrobial drugs that placed the majority of my symptoms and conditions into remission. And I thought to myself, okay, I'm better now. I previously worked for the world's largest pharma company, Pfizer, then the world's first biotech, Genentech. So I knew how to navigate the medical literature, but I felt that that was extraordinarily unfair that a patient like myself, like how many other young mothers with kids ages 3 and 5 are lying down on the couch somewhere thinking they're dying, with terrible diagnoses in their differential, but have no answers yet and may have a treatable infection. It turned out that not only did I have the chlamydia pneumoniae bacteria that had likely been involved in lifelong, very severe asthma, but I also had Lyme disease, a bacterium and set of organisms transmitted by tick bites. I had been living in Connecticut and was bitten and contracted Borrelia burgdorferi, the organism that causes Lyme, but I also got another bacteria called Bartonella henselae, which we are realizing is far more prevalent and far more of an issue than we originally thought with our research, and a parasite called babesiosis, which had actually produced very unusual symptoms for me, like air hunger. Literally, I couldn't get a breath. And so, fast forward to today, the Pathobiome Research Center is really both the vehicle and, the result and the future for what Brian and I collectively, with a global multidisciplinary team, have essentially decided this is a bigger problem than any one single bacteria, virus, fungi. This is a bigger problem that we are overlooking. We know infectious diseases can cause chronic diseases. There are so many examples, right? HPV can cause cancer. There's a stealthy bacteria that causes stomach ulcers. Human immunodeficiency virus, HIV, can lead to other infections and cancers by suppressing the immune system. So it's a well-established paradigm, but we haven't applied it in other places. And so this global team of ours and Brian's lab, the Center for Chronic Disorders of Aging, which is actually conducting the research looking at brains from patients that we lost to these devastating diseases and searching for microbes and also looking for other factors in the lab, collaborates closely with my collaboration hub, which is a really unique concept. The Pathobiome Research Center isn't a wet lab. It is a hub for building collaborations that are multidisciplinary and with other universities and centers. And in so doing, we've been able to bring together the world's leading scientists that are studying different aspects of this problem. But the general idea of what we do is how do infections impact chronic disease? And specifically, we've focused in on the brain.
Well, it's a great history to get us to where we are. Brian, you're like the classic story of an actor, who breaks it into onto the scene and wins an Oscar, and that no one never heard of, but has been acting for 40 years.
That's about
you
right.
you know you're getting you're getting your day in the sun right now. So how do we get this into mainstream treatment and prevention and start moving off these long accepted theories of amyloid that aren't panning out clinically from a treatment standpoint? How are we going to bridge that divide and start convincing physicians in the clinical world they need to look at things differently?
What's really interesting here, Jay, is that, and I've talked to clinicians about this, and we have some clinicians we're working with at PCOM, our geriatricians, Dr. Galuzzi, Dr. Joseph, for instance, and we've compelled them to start actually testing differently with individuals coming in with memory complaints. One of the testing differences here is for smell testing, is to see if there's any difference in our sense of smell, which we call olfaction. And I mention this because I've talked to a lot of other clinicians, medical students, etc., and they all tell me they never test routinely, even ENTs, for change in sense of smell as cranial nerve 1. They test cranial nerves 2 through 12, but we have 12 cranial nerves. Why aren't we testing for cranial nerve 1? And I say this because it's been recognized in Parkinson's disease, in Alzheimer's disease, in schizophrenia, in autism, when COVID hit, that the sense of smell was impacted. And with all of these conditions, it tells us that olfaction is a key to the problem. We have a keystone. We need to focus on why is that a keystone? It is the easiest access to our nervous system beyond any other types of access into our bodies, of something to go into our brains, to impact now the areas of our brains that are always affected, and I'm saying always, in neurodegenerative processes, which are extended beyond Alzheimer's or Parkinson's. We're talking about, and schizophrenia, we're talking about major neurodegenerative processes like frontotemporal dementias. Pick's disease is part of that. Other types of changes in our brains that are connected to olfaction. It's almost as if we've lost the sight of the forest through the trees. It is straightforward. There are smell tests that we can use routinely. We need to focus that way. And then we need to sample better. We can do nasal swabbing. We can do saliva collections. We can do blood collections, urine collections, etc., to find what's on board in an individual to better diagnose by looking for an infectious process, which we believe would be there and is there in maybe many different ways, different types of infections, that we could now then start doing something about through a treatment of antibacterials or antivirals, antifungals, antiparasitics that may be playing a major role in the different populations. So this polymicrobial issue is really what we're talking about here, and it extends even from the nervous system to the rest of the body. We are a whole unit. This is why at PCOM, an osteopathic facility, an osteopathic school, it is the right way to study the problem. And this it gives us the advantage of looking at the whole person. How are we being affected this way? And this is what Nikki's work really is designed to do with the Pathobiome Research Center, is to expand this to over the lifespan, in fact, as well, to how chronic infection that we encounter can actually impact us from birth to even before birth and congenitally to death.
Yeah, and Jay, to go back to another point, and Brian, thank you so much for raising all of that. One of the main goals and missions of this Alzheimer Pathobiome Initiative Consortium that Brian and I co-founded alongside others, including Dr. Garth Ehrlich at Drexel, so we have a strong Pennsylvania presence, and Dr. Or Shemesh at Pittsburgh, is to develop what we call a consensus testing approach. So what we want to know as a team, and we're trying to answer scientifically, is what test would we give someone? Let's say, how you go get a mammogram at 40 if you're a woman, and you go and get a colonoscopy at 50 to look for issues. Being that we know that infections can indeed cause dementia, and there are ample examples of that in the literature, and different clinical entities, it's very logical to ask if the most common form of dementia, Alzheimer dementia, is being driven by infection in certain cases. And how would we know is by screening and testing. And so, one of the things our team is attempting to answer scientifically is we know that a lot of the microbes we study are stealthy, they don't want to be seen. So, using typical antibody based tests that look at the patient's immune response to the infection is not the best way to do it. It may be informative in conjunction with other tests. So, what we would like to learn about is, comparing specimens from the same patient. And, unfortunately, testing patients that we already lost to these devastating diseases by looking at what happened in their brain in the regions where the disease was, and also looking at their blood and things like spinal fluid and saliva, looking for different pathogens, comparing them. If we start to see a signature and notice that we can detect these things, let's say in saliva, that would lead to a testing panel approach that could be implemented early in life because we note that infectious syndromes look differently across the lifespan. We also study, as Brian mentioned, pediatric disorders and brains from patients that were lost to those neurodegenerative and neuroimmune diseases, because the way that a child's brain responds and a child's immune system responds to the same stimulus, the infection, is very different. But it can be informative and tell us about clinical signs and symptoms that predispose one to later in life developing a devastating neurodegenerative disease. It also tells us about shared mechanisms and allow us to really remain broad in terms of our purview of what we're looking at. Because one of the things that we know about innovation is that innovation doesn't happen most readily in a vacuum. It happens by bringing multidisciplinary groups together that pressure test assumptions, and that's exactly what we're doing. Jay, earlier you mentioned, removing pathology, like we have drugs that can clear amyloid really well. Well, they're not incredibly effective or incredibly safe. So we know that we need to be doing more. We need to be thinking upstream of what triggers those things. Multiple of our Alzheimer Pathobiome Initiative and Pathobiome Research Center collaborators have demonstrated that those hallmarks of the disease, amyloid and tau, are innate immune defenses, meaning that those are the firefighters showing up to combat a process that's occurring, whereas the field has mainly understood them as arsons. They're there with the match lighting the fire. Well, we are really looking upstream.
How far away are we from the tests that you're talking about that can then be translated into clinical care and trials for antibiotics for certain chronic neurologic conditions?
Well, we already have tests. Unfortunately, many of the best tests are not currently covered by insurance. Like Brian, we've written grants to go into the federal government, leveraging companies like Novogene, for example, to do something called sequencing, which would tell us about, your DNA. Everything has a map of the directions for the body, right? So a human's body, we all have every cell in our body has a little map of how to make that entire body, which is fairly incredible. Your DNA, your building blocks. Well, pathogens too, we can look for their DNA or their RNA, and meaning essentially that they're alive and well. They're in that specimen by and large doing, something. They're active, they're alive. We could leverage these tests that are already on the market, but the challenge is for the clinician practicing is that these will not be covered, not used in that capacity, but we have the capabilities already. Now, the point of us doing this research project that we're doing is determine what is the best specimen, right? One of the points of the project. And then also, which are the pathogens that are triggering pathology. And we note that many different pathogens can do this. So it's a disease pathway where we really need precision. And that already exists. Like this concept is uh, very well accepted in infectious diseases, right? If someone has suspected meningitis or encephalitis, they'll be in the hospital. They may start antibiotics empirically before they know what's going on, but they will do testing exhaustively to determine what's going on in that patient and make sure that the drug actually kills what they find, because some of these pathogens can resist the drugs. So we not only need to find out what's there, but if they're vulnerable to the treatments that would be given. And so what stands in our way really is a grant that's large enough to enable us to use the platform and team we've already developed. We wrote a plan back in 2023 to go after this issue using a global team of experts and using multiple different ways of interrogating this problem so that we can show which germs show up, how do they do their dirty work, right? Mechanism, and then what testing approaches would we use on what specimen so that we could say at the end of the day, now LabCorp has a test and it is going to be a mixture of blood and you're going to spit in this tube and it's going to use X, Y, and Z assays. And this is your risk score for going on to develop a neurodegenerative or neuropsychiatric condition. That would be the dream. It's not unattainable. It just needs to be funded. And federal funding has been a great challenge for any paradigm shifting ideas over like this. Right, Brian?
Absolutely. That is a big roadblock for us. Part of this is because with the approach we take for funding at the NIH is that it goes to study sections of individuals that typically are not studying this problem. And so they come up with different types of actually arguments that really are not very accepting of even what we're proposing here with data, with the evidence we have. For example, we have recently been criticized that potentially what we're finding with infectious agents in brain tissues of people with Alzheimer's disease is because they already have the disease. Well, that's an archaic way of thinking about the whole problem, that the disease is allowing for infectious agents to come into the brain. Actually,
Wait a second, let me back up. So the reason they have the infections is because they have Alzheimer's?
yes. So it comes down to this, is that you would think that based on comments like that, that we knew what causes Alzheimer's. We accept what causes Alzheimer's disease, which we do not. In fact, the exposome has become a very popular term now. And now people writing about an exposome, they talk about 14 different factors, leaving out infection as being one of those exposures that could lead to the disease, which is remarkable to me. I've been doing this for so long, Jay, as you mentioned. It's remarkable to me, even after COVID. And now, even during still sequelae from current COVID cases, COVID, the SARS-CoV-2 virus, enters into our brains through the nasal passages, through olfaction. It has led to smell loss in many cases. It has led to memory changes, cognitive deficits, long COVID, brain fog. Okay? It's a perfect example of a respiratory organism that can lead to sequelae. A recent study just came out in Nature, the periodical Nature, demonstrating that people that got COVID actually had reactivation of other viruses that started to become in immunocompetent people, meaning their immune systems weren't compromised. They were like normal people that got COVID, and now you had reactivation of Epstein-Barr virus, cytomegalovirus, herpes simplex 1 virus, and another type of virus called Anelloviridae. Well, these viruses are on board in us commonly.
Right.
So here's one infection that led to others being reactivated. So we think that may be happening also with other organisms that we're looking at, that it could lead to that, a polymicrobial insult. But again, olfaction is the key. And I keep focusing that way because it's something that we need the public to know, the other researchers to know, etc. Our research world, our other scientists that evaluate the work we're proposing are not capable of understanding the paradigm shift that we're talking about here. We're talking about quite possibly, this is the keystone, infection as a keystone to causation of these major problems.
So we talk about the word exposome, meaning like everything that hits you over the course of a lifetime. these things can actually, they're not unrelated to infection. So it was cool. I believe it was Dr. Ken Blank commented on one of my educational videos that I'm creating, and it can be followed, by the way, on Instagram and on LinkedIn and all the social media platforms with credible information in digestible sound bites about these things. But, Dr. Blank said, well, don't forget about genetics. So inherently, when we're talking about infections, we're also talking about genetics and a bunch of other things because this is all a web. They're not disconnected from each other. So our genetics have been formed in many ways from pressure exerted on human species over such an expansive amount of time. One of the number one sources of pressure were infections that we had to survive. So a cool thing that most people don't realize is the Alzheimer's gene. People are aware there's a genetic predisposition with something called ApoE4. It's all capitals and then the number 4. If you have 2 copies of that gene, you have a extremely heightened risk of developing Alzheimer's disease by a certain age. It can be modifiable with certain lifestyle changes. So it's not necessarily at this point in time something that you can't do something about it. But one of the things it does is enhance the ability for some of these infections to get into our cells, which gives them access to our brain. And then it also increases, scientists like Brian, Dr. Ruth Itzhaki, others have shown that it can increase the load of infection in the brain of patients that we lost to this disease, meaning how many bacteria, viruses, and so forth. But it's not all bad. So you say to yourself, why would we still have this gene if, Darwin and natural selection, it's happening, right? Why do we have the ApoE4 gene? Well, it's ancestral and it's been around the longest and it protected our ancestors from malaria.
Sickle cell is a gene to protect, people from malaria as well.
Correct. Very
Understanding
true.
points of vulnerability, we can't, we could, there is gene editing now, but I don't necessarily think that's the answer. I think it's understanding what the individual person's vulnerability is and then acting upon that. There are nutritional deficiencies, hormonal issues that we realize are also important. So we're not just saying infection is the only thing. Air pollution and what you're exposed to, heavy metal toxicities. There are a lot of things to consider, but this is one of the big things that's sort of been overlooked. And it hasn't been profitable, really, to go down this road. The pharma industry knows that antimicrobial, anti-infective drugs don't bring the return on investment that other drugs will bring, like biologics or rare disease products. So there's been a general lack of economic stimulus for this. But with me knowing what it feels like to have neuroinfectious involvement and knowing that other people out there are going through this and they don't have a diagnosis drives drives our team every day, right, Brian? I mean, we are constantly thinking of ways of raising awareness, raising research funding, and getting really paradigm shifting work in a light where it needs to be seen to be regarded.
We're compelled to have this happen now. And the reason being, Jay, is that in the US alone, 120,000 individuals with Alzheimer's disease are dying on a yearly basis. Over the course of the world, where we have 57 million documented cases, 2 million are dying annually from these neurodegenerative dementing issues. We are compelled to make the difference now. And for the Alzheimer's Association, the NIH, Big Pharma, other organizations that keep pushing away from this, keeping this at a distance, is a disservice to humanity. And we're not doing this because we think, oh yes, we're going to get the acclaim for discovering or finding or determining how the disease actually is occurring and what's causing it. Is to make the difference for the population. We're part of the population as well. Okay, so we could say, well, yeah, it's a selfish thing on our part as well. But guess what? We're trying to make the difference for humans, for anyone that is running the risk here of getting these diseases. And for this to be denied, this not to be covered, this not to be investigated is a complete disservice to us as a human population.
the passion that you two exhibit for what you're trying to do is palpable. And I think it's a credit to the both of you. And what you're doing is really trying to change the course of a disease for the benefit of humanity. And, I want to thank you both, for being here today, for joining us, for sharing your insights. And your interdisciplinary approach not only advances important research, but also creates meaningful hands-on opportunities for PCOM students that can shape the trajectory of their careers and the trajectory of humanity. So we really appreciate the impact of your work and look forward to seeing what's ahead for the Pathobiome Research Center. And I'm Dr. Jay Feldstein, and this has been PCOM Perspectives.